Additive antitumor effect of arsenic trioxide combined with intravesical Bacillus calmette-guerin immunotherapy against bladder cancer through blockade of the IER3 and RF2 pathway by Isabella Buitrago Medina.
The cells in our bodies all have certain jobs to do. Normal cells divide in an orderly way. They die when they get too old or damaged, and new cells take their place. Cancer begins when genetic changes interfere with this orderly process and cells start to grow out of control. These cells may form a mass called a tumor. A tumor can be cancerous or benign. The immunotherapy, also denominated biological therapy, is a type of treatment for the cancer that stimulates the natural defenses of the body in order to attack the cancer. It uses substances produced by the body or made in a laboratory to improve or to restore the function of the immune system. The immunotherapy can work of the following ways: can mark the cancerous cells in the sake of facilitating the immune system to find and destroy them, or immunotherapies reinforce the immune system in order that it works better against the cancer, or to delay the growth of the cancer cells or prevent the cancer to spread to other parts of the body. Arsenic trioxide is an inorganic compound. Even though it's recognized because of its toxicity, it can induce cellular apoptosis in certain types of cancer, including bladder cancer. Intravesical Bacillus calmette-guerin is a live attenuated strain of Mycobacterium bovis that is applied as a vaccine against human disease. It could be an intravesical treatment option for non-muscle invasive bladder cancer. Both work together to counter cancer, as arsenic trioxide disable the root IER3 and RF2, and that BCG promoted expression of interleukin 6 and interleukin 8. Objective: the aim of the present study was to elucidate the crucial cooperative role of tyrosine and intravesical Bacillus calmette-guerin immunotherapy and its ability to protect against bladder cancer by targeting the IER3 and RF2 pathway. Methods and materials. Reverse transcription, quantitative polymerase chain reaction. It calculates the relative expression levels of genes. It means that it quantifies the RNA that has been transcribed in a biological sample in order to detect, characterize, and to quantify nucleic acids. The mold that is used for the quantitative PCR is not a DNA from the beginning. Instead, it's a complementary DNA of sample fiber obtained from a transcription of ribonucleic acid. Since the conventional PCR, the DNA is amplified by following 3 repeated steps. The first one is denaturalization of the double DNA chain. Then, the union of the primers which are IER3, NRF2, and Q01, interleukin 6, A, and B acting to the DNA simple strand, and the last one, stretching of DNA's chains mold. The test was used for being able to see if it had or not the gene duplications or deletions in the different target genes, since these serve as cancer markers, such as NRF2, that is a promoter of tumors. Western blot is often used in research to separate and identify proteins. In this technique, a mixture of proteins is separated based on molecular weight and thus by type through Hall electrophoresis. The results are then transferred to a membrane producing a band for each protein. The membrane is then incubated with levels of antibodies specific to the protein of interest. The unbound antibody is washed off, leaving only the bound antibody to the protein of interest. The bound antibodies are then detected by developing the film. As antibodies only bind to the protein of interest, only one bound should be visible. The thickness of the band corresponds to the amount of protein present. The main purpose of this method was to give an idea of the total proteins in the dendritic cells of mice that can codify the information of bladder cancer and the possible post for the formation of bladder cancer and the possible post-translation modifications. Immunohistochemistry. It is based on the evaluation of the reaction between an antigen and the antibody. In this case, the reaction is demonstrated in a tissue and it can be demonstrated by a marker that then is evaluated to know if it is positive or negative. Immunohistochemistry was used in order to analyze the antibodies S100 to mark the dendritic cells, since these are capable of synthesizing and secreting the above-mentioned protein. In addition, they are antigen-presenting cells. Among the causes responsible for the insufficiency of the immune system to stop tumor progression is a defective antigen presentation. ABC ELISA. The basic principle of ABC ELISA is to use an enzyme to detect the antigen-antibody binding. The enzyme converts a colorless substrate, the chromogen, to a color product, indicating the presence of antigen-antibody binding and leading to know the concentration of an analyte in solution. This method was of great importance due to the fact that it showed the presence of interleukin-6 and interleukin-8. This will be of great help for the diagnosis of bladder cancer since the interleukin-6 intervenes in the production of immunoglobulin and the differentiation of lymphocytes B. Results figure 2. Okay, so in this figure we can see how trioxide arsenic and BCG increase expression of CD83, CD86 in dendritic cells. We can see the changes presented in dendritic cells from the first day up to the 7th one. There are a lot of morphological changes, for example, in image B. The cells are spherical, the protuberances in the majority of the cellular surfaces are stretched and they get thicker. Also, the volume is bigger compared to a lymphocyte B. Okay, so here we can see how it increased the CD83 and CD86 with trioxide arsenic and BCG. Results figure 3. So here we can see the protein expressions of IER3, NRF2, and NQO1 in dendritical cells after different treatments determined by Western blot. This is a result of Western blot trial. DB-actin is the test control. These 3 proteins were evaluated and their expressions in these different compounds. In all of them, we could see that the reactions are positive, but in these ones, this one and this one, it decreases its expression of the NFR2 and the NQO1 proteins. Therefore, we can deduce that these compounds are not expressing this protein, like this one, the NRF2 protein, which controls the way in which certain genes are expressed, which help protect the cell from the damage caused by free radicals, are those that are most related to bladder cancer. Additionally, we can see that the trioxide arsenic group had lower expression levels of IER3 but higher expression of NRF2 and QO1, interleukin-6, 8 than the control group. Okay, so in this figure we can see the number of dendritic cells in these different compounds. The average amount of dendritic cells in the bladder tissues was higher in the BCG and trioxide arsenic groups than in the blank group. There was no significant difference in the average amount of dendritic cells in bladder tissues between the blank group and this group. Compared with the BCG group, the trioxide arsenic plus BCG group had a higher average amount of dendritic cells in bladder tissues. However, compared with the trioxide arsenic plus BCG group, this group had a lower average amount of dendritic cells in bladder tissues. The size of the dendritic cell in the first image and in this image is smaller than in any other of the compounds. It can be observed that it always maintains an intense basophilic color except in this image, that the color is quite clear. In addition, the shape of the cell stays round except in this one, that we can see that it's a little bit elongated shape. Okay, so this figure shows us how trioxide arsenic cooperates with BCG to promote apoptosis of tumor cell and inhibit tumor progression by downregulating the IER3 and RF2 pathway. It can be seen that in trioxide arsenic with BCG is in which there is the greatest apoptosis index, which is what is needed to treat cancer, while the most diminished is in this one. On the contrary, it is seen how the volume and weight of the tumor is lower in trioxide arsenic than in any other of the compounds. Discussion. So, these 3 authors say different things. So what they propose, the key evidence presented in our study disclosed that trioxide arsenic and BCG were able to promote the apoptosis of bladder tumor cells and decrease the volume and weight of tumors. The pro-apoptotic function of trioxide arsenic was a significant finding that was noted. BCG has been shown to stimulate the increase of CD4 cells. The efficacy of BCG has been associated with the increase of dendritic cells. During the presentation of the entire study, these 3 aspects were demonstrated and achieved. It was seen how the joint work of BCG and arsenic increased the rate of the apoptosis necessary for the death of cells and fighting cancer. In addition, the increase of both dendritic and CD4 cells whose function is given through cytokines to enhance the immune response, was very high thanks to these compounds. Conclusions. Increasingly, researchers are striving to find different ways to prevent and treat cancer. It is of great importance that this type of research continue developing as more people are affected every day. Mainly, this research helps to pose as how a toxic agent and an inactive form of the bacterium Mycobacterium tuberculosis can generate many effects to counterattack the cancer. More research should be done with innovative foundations to be able to help the field of medicine to answer all of the unknowns about cancer and be able to give a better diagnostic to fight cancer.